All right, we're going to talk more about DNA technologies, biotech, and today we're going to focus on using DNA polymerase. That's an enzyme that copies DNA. And we're going to describe how DNA can be copied, how genes can be copied, sequencing, and I'll talk a lot about DNA profiling. And so these are all technologies based on using enzymes and test tubes and automating it. And that picture you see there, that's a picture of a hot spring. And that's where we got this enzyme that from a hot springs organism. Okay, so last time I presented a little uh, picture of the one of the first biotechnologies kind of understanding DNA, and this was the cloning of an organism. And that was really simple technology. You just take an egg cell, remove the nucleus, and insert the nucleus from an adult cell. And so that was the um, um, an adult cell from a mammary gland. You put that nucleus in by fusing the cells, and then you stimulate the cell to divide and implant the fertilized egg in a host, and you've cloned an organism. Not very difficult to do, easy. Really working with a pipette and a needle and a small electrode. Okay, the other big thing I introduced was cloning and creating recombinant DNA and recombinant cells. And here we use a pair of enzymes, a restriction enzyme that cuts DNA, and then an enzyme that heals the cuts. And so we can remove genes that we like, cut them with one enzyme, and put them in a piece of DNA that it's easy to insert into a cell by healing them with DNA ligase. This is how we produce insulin. It's how we make recombinant cells. Um, we produce many medicines this way. We've put genes in plant embryos and grown plants that are recombinant. This is big, big money, and it saves a lot of lives. Very valuable. Um, Not all DNA technologies have lived up to the hype. I want to mention one that's only now starting to be used, even though it was one of the first ideas mentioned. That was gene therapy. This was inserting a correct copy of a gene that was defective in somebody who had a genetic disease. And so we would replace the damaged copy with the correct copy. So this was attempted for cystic fibrosis and muscular dystrophy, and it is only now starting to see some limited success, but it's been tried for over 30 years. Um, you've got to be able to get good genes into the affected cells. This has turned out to be very, very difficult. Um, if you want to cure muscular dystrophy, you have to get a correct copy of the gene into all the muscle cells and not into any other cells. So that's been really, really difficult. Okay, so today we're going to talk about DNA. It's a double-stranded molecule made of 4 nucleotides. All cells use DNA. All cells have the same base pairing, with cytosine pairing with guanine, adenosine, adenosine pairing with thymine. All cells have the same genetic code. It means use the same enzymes to do this. And so this has been really an important realization that we can do this anywhere. Also, we can find specific genes by making probes that bind to specific sequences. And we can use enzymes that do this and start the enzymes off even if they need specific primers. And so we have the things to use DNA and to get it into uh, to copy it. And the enzyme that does this is DNA polymerase. And one of the great kind of advances is that we're able to copy DNA. And we do this through the polymerase chain reaction. So we use the enzyme Taq polymerase, which comes from a bacteria Thermus aquaticus, and it's a DNA polymerase in a hot springs bacteria. So the enzyme is heat stable and actually likes working at temperatures that are very, very high, that virtually kill almost all other bacteria and contaminants. All this enzyme needs is a primer, some raw nucleotides, and some source DNA. And then a machine called a thermal cycler, which cycles between temperatures that cause DNA strands to separate and temperatures that the enzyme likes to work at. And that sounds like it's really hard to get, but this is just a computerized hot plate. And so the picture here shows the thermocycler, the bacteria on top in purple, and then the hot springs where they came from. So what happens in PCR is the thermocycler goes to 95 degrees, and that causes double-stranded DNA to separate. Then it's cooled down to 50 degrees, where primers bind to the DNA, and the DNA enzyme, DNA polymerase, attaches to it. Then at 72 degrees, that's the temperature the enzyme works, and the DNA is copied. Okay, and so you go to 95 to 50 and to 72, and the DNA is copied within a few seconds. Then you cycle again back to 95 to separate the new strands, cool it down to 50, RNA primers bind, and then you get it to 72. And so you double the number of strands with each cycle. And in 30 cycles, you can produce billions of times more DNA. Okay, and that all happens 30 cycles you can run in 3 hours. And so this is automated, and you can produce unlimited amounts of DNA from any sample, from the environment, from a single cell, anything. So we have lots of DNA to analyze. We also use DNA polymerase in another automated process, automated DNA sequencing. Here we use source DNA, an enzyme, DNA polymerase again, and then primers and nucleotides along with something called chain terminators. And this automated sequencer again cycles between temperatures. Okay, and here what happens is these chain terminators are labeled with a fluorescent dye, and the different uh, there's 4 different ones, one that resembles thymine, one that resembles guanine, one that resembles adenine, and another that resembles cytosine. These chain terminators, they will stop the copying of the segment, and at that place where they end will be a fluorescent dye. So you'll know how long each segment is because you can separate them by weight, and then you'll know the sequence at the end because there's a fluorescent dye. So this machine is really easy to operate. You just load your samples. Again, the DNA polymerase works on cycles. You have nucleotides and chain terminators, and then you separate the fragments by size, and the computer looks at uh, what dye is at the end of each fragment, and it just reads it. And you can sequence a whole genome in just an afternoon. Or in some cases, you may just do parts of the gene, and you can recognize this. Okay, the application I want to talk about that is DNA profiling. Okay, we used to recognize people by blood type. Okay, so the only biological sample that we could identify reliably was different blood types. And there's only 8 blood types, and even the rarest blood type is about 1% of the population. So the best scientific evidence we had to identify individuals were these blood types, and often you could only narrow it down to half the population. With DNA profiling, we use genetic techniques, polymerase chain reaction, sequencing or matching, that allow us to identify individuals to very, very fine levels, 1 in a billion or more, so that we can say very conclusively this sample came from this person or their identical twin. And so there's these DNA profile techniques include identifying variable regions called short tandem repeats. Frequency, we can cut the DNA in samples and measure what the cuts are, or we can actually sequence it. But we can use extremely small biological samples from blood, hair, saliva, semen, or even skin cells, and we can identify them extremely specifically. Okay, this has led to some really important effects on our justice system because now we can identify people much more precisely and we can rule them out much more precisely. And so evidence that was used, biological evidence that was used to convict people, is now being used to exonerate them. And so there's a project called the Innocence Project that's was started by a group of lawyers to re-examine biological evidence using DNA technologies. This has overturned over 375 convictions for people that were often on death row or facing life in prison who were wrongly convicted. Often, this same evidence that was used to convict them with DNA technologies can be used to find the real perpetrator. And so these overturned convictions often lead to new convictions. Um, for example, I'll give you Illinois' experience. Um, we we had in between 1977 and 2000, we executed 12 people in Illinois. But with between 1990 and 2000, we started relooking at those people who were convicted of these capital crimes with DNA technology, and we overturned 13 convictions because the evidence clearly indicated these people were not matched with the biological sample. That experience led the governor of Illinois to put a moratorium to ban executions because it was clear that lots of convictions were in error, that people were convicted based on evidence that was weak and could be refuted with new scientific evidence coming up. I'll show you some film clips from this, but it's really amazing, this detail that we've gone on. So in summary, there are techniques that we can use to make unlimited copies of any DNA sample. We can find any DNA sequence and we can determine the base sequence of DNAs. This is really important applications for our criminal justice system. It's allowed us to really refine what we've got and to see some of the errors in the justice system that we've before. It also has other applications in finding genes and relating organisms together that I'll talk about later. Okay,